without any further ado, I think we'll turn it over to you guys. And again, we see this as being somewhat informal, just letting Lisa and Diana do a little bit of an overview, and then just opening it up for questions, any questions that folks might have that we can, bring out and discuss here. We are recording this, so if anyone has an issue with that, please let me know. And if not, we will make it available with the series as we continue. Our CMC video is now up on our YouTube channels. Okay, great. Lisa, I will turn it over to you.
Thanks, Heather and Logan, and thanks so much for inviting me and allowing me to include Diana. I feel like the folks on this call know a lot of information on these topics, both regulatory and clinical trial issues. So I'm very happy that Diana was willing to join me, because I think many of the questions are going to be about investigator responsibilities and sponsor responsibilities, even outside of regulation. But I'm going to start by quickly introducing myself for those on the phone who may not know me. I've met many of you, but I'm an OBGYN here in the Washington, D.C. area. I'm not in practice. I worked at the Food and Drug Administration for 15 years, all in the women's health space, and most notably in the division that handles the reproductive health drugs, including drugs that would be used for male contraceptive methods. And we've been looking at these things for a long time, and I'll bring some of those issues that have been hurdles into the talk at the end of my presentation. I've been consulting to pharmaceutical companies since about 2003, and I'm really happy to be included in the discussions and advisory context to the Male Contraceptive Initiative. Thank you, you all. So Diana, if you'll introduce yourself, then I'll go through what we're going to talk about today.
Hi, I'm Diana Blithe. I'm Chief of the Contraceptive Development Program at NICHD in the NIH. And I've been working in this field now for a little over 20 years, developing female methods and male methods. We've actually gotten some female methods all the way through a very long pipeline to approval. We're hoping someday to get a male method approved. And I'll talk a little bit more about our experience with that to date at later point in the talk. And
thanks, Diana.
I hope you'll forgive me, because I am not as technologically savvy as many of the folks who are on this call or elsewhere. So thanks.
Well, as folks will realize when they see my PowerPoint slides, I have no technological savvy. These are your basic government type of black and white PowerPoint slides with very skeletal topics. I also wasn't sure of who would be included in the conversation. And as I said, many folks on the call have had various experiences dealing with the FDA, and I look forward to hearing their comments about their knowledge and expertise as we go through these topics, and especially hopefully at the end when we have some more open discussion. As you see, we've already introduced ourselves. I want to make clear that my experience has been in the pharmaceutical drug side. I know the only male method right now is condoms, which are considered devices, although they do have non-oxidil 9 on them, so we can discuss maybe if there's some future for a better lubricant or spermicide products with a condom. But I personally don't have enough device knowledge to give you much help with submitting IDEs or investigational drug exemptions or PMAs or 510Ks to the device group. So I hope that Logan and Heather will consider engaging one of the device experts to have that discussion with you at some future lemonade stand discussion. And I'm going to simply go through some CDER regulations and guidances that exist that I think are really important to any kind of startup or anybody interested in these topics. And then I will end my part with the kinds of obstacles that have really been a struggle for companies or for investigators thinking about this space. And then as you see in the red, I will turn it over to Dr. Blithe for the challenges and the discussion of product development and timelines. So in terms of FDA requirements, I would really encourage everybody to just simply go online to search for 21 CFR. It's called the Code of Federal Regulations, and look up 312. When I first joined the agency, I remember I was asked, that was back in the days where we could actually go to make presentations, and I made a presentation to Serono, of all things, in the late 80s and early 90s, and I was asked to discuss what's in an IND and what's in an NDA. And so I discovered very early in my career, as all regulatory people should know, the CFR provides incredible information, and 312 is the one that guides and regulates INDs. And it's not, if you click on 312, it's a 2-page list of a topic, the table of contents, and it provides lots of information about whether or not you can promote, whether or not you can charge for your investigational product, describes the requirements, the phases, how to submit, how to do safety reports. It describes clinical holds and disputes and meetings, and as well as your responsibilities. It describes when you can include foreign studies that aren't conducted under your INDs, and it describes that you can find guidance documents. And there really are a tremendous number of printed guidance documents that are available on the FDA's website. You go to the drug site, go to guidances, throw in a keyword and something like investigational, and you'll find just dozens of drug development guidance documents, including answering questions like, when do I not need an IND? What's exempt? When can human studies be done without an IND? And it goes through all the things that you probably already know. If you're going to exempt, it has to be an approved drug. It has to be not intended to be reported to the FDA as a well-controlled study to support a new indication. It's not going to be used to make significant changes in advertising, and it doesn't increase the risk to the patient population. And there's guidance that discusses all of those aspects of when you don't even need an IND. And then there has been some confusion about phase 1 studies and how complete a submission needs to be, especially in terms of the pharm-tox studies needed, the pharmacology and toxicology studies needed to do various types of human trials. And there are guidance documents both for exploratory IND studies. Essentially, those are usually single-dose studies where animal studies may only be a week long, but they support a single dose for simple questions such as pharmacokinetics, et cetera, not necessarily dose finding, but simple questions. And the pharm tox requirements there are quite less than for a true IND. But again, there's a guidance document for the entire IND and what really needs to be there from chemistry, from pharm tox. As you'll see from the guidance documents for phase 1 studies, they're really about CMC and pharm tox studies, and just a very short discussion of what needs to be provided from a clinical aspect. And oftentimes it's a simple synopsis that we'll do with some sense of numbers of subjects and length of study to go ahead and get an IND up and running with the agency. I do get questions about charging. There are guidances about that. Lots of questions about doing foreign studies. Foreign studies can be done without an IND, but if the study is expected to be submitted to the agency for any sort of approval action, those need to be done consistent with FDA guidelines and guidances and requirements. And there are positives and negatives to doing all completely foreign studies without an IND versus with an IND, and those kinds of questions we can talk about if you'd like. There's a guidance about having meetings. Many of you are aware of having meetings with the agency. Obviously, right now, they're all doing written responses. There's no face-to-face meetings currently, but I find written responses from the agency actually pretty useful. I think, although meetings have a benefit of sort of that personal connection, I think the agency is more willing to state things in black and white when they don't meet with you. And so you can get a little bit, I think, clearer direction in a written response. If you get put on hold, there are guidances for what you have to do to get off clinical hold, as well as timeframes for how that works. Safety reporting is something that a lot of smaller startup companies may not be completely up to speed on, and I encourage folks to read guidances regarding what is considered serious and unexpected adverse reactions, and what can be provided in more of a comprehensive way at annual reporting. And there are data retention requirements by the agency, as you'll see if you read the guidance documents. See, I'm going to whip through this. I really wanted to just get to some common issues, and these are really broad, but I'm constantly reminded that these male contraceptive methods involve safety for 3 populations, and what a challenge. You know, it's not just the short and long-term effects in the male. It's also, well, does it have any effect in the female? And then if something fails, does it have any effect in the fetus? And these are kinds of questions that can be very difficult. I remember a sperm vaccine during my tenure as a reviewer that just simply couldn't get through the hurdle of, you know, creating antibodies in the woman against sperm, but not being able to really answer what happens if she's then pregnant with a male fetus. And it just such a challenging space. And so we have to think down the line, you know, what is the agency going to be expecting a sponsor to answer, and be sure that if there's any way to use a preclinical model, make arguments about placental transfer, and then figure out if there is a failure and a pregnancy, what the sponsor is going to do regarding that pregnancy and how to follow it and how to assess long-term safety. So as I mentioned, subject safety is all of those 3 populations: women, the men, their partners, short-term issues of acceptability, and long-term risks. And I'm going to stop there. If somebody wants to ask me questions quickly, I'm going to stop sharing. It'll take a second for Diane to pop hers on. So let me stop there and let you ask me any questions. Not hearing me. I see a lot of people are muted. Okay, go ahead.
Okay, so I'm wondering whether you could just elaborate a little bit more on you know, the contraceptive failure and subsequent pregnancy. You know, do you have some, I guess, more information on that? What, you know, one would monitor, and I guess, I mean, it's a real, you know, could be a real problem.
Well, I think it really depends on what your method might do to sperm, and what that sperm change might do to a pregnancy. Right. In many ways, in many times, you're going to argue that if someone gets pregnant, then it didn't affect the sperm, or, you know, it didn't. But you're going to have to make that argument, because if there's any question that there's something that has altered the health of a sperm, and yet someone gets pregnant, you know, what does that going to entail in terms of safety? It's going to be very case by case in terms of trying to answer that question. You know, if the method is such that it's affecting, you know, I mean, Diana will have a better idea of the basic sciences, but if it's affecting something that makes sperm is perfectly healthy, but for some reason is unable to you know, fertilize an egg or something, then it might be safer, because if a healthy one gets through, a healthy one gets through. So it's all going to depend on what happens, you know, what's going on with that method, and if it's sperm specific or environment specific, to try to understand what risks that entails.
I mean, I think the point you're making is, of course, that, you know, if a sperm is sort of damaged by, you know, I guess whatever measures that you have inhibited certain processes in spermatogenesis. So, I mean, that's a good thing to think about, and I think that's a reasonable assumption. But, you know, what happens if there is a slight damage, right? And so I guess that's sort of the issue that we have to kind of be aware of, right?
Right, and I think you would be responsible to answer that. I mean, that's the problem. There's going to be this sort of unknown, scary risk, and you guys are the people. You guys are the scientists that are going to put together the argument about why that's okay.
Lisa, just practically speaking, aren't you speaking to the length of follow-up that would be required in any human introduction? I mean, for women, if you're talking about long-term effect and not knowing what that is, and you've got to consider what happens in a pregnancy, that means you're going to be following women for a long time, or the partners of the men in the trial for a long time, which just leads to another set of complications in that filing, I would assume.
Yes, exactly. I mean, there is a guidance for post-approval risk assessment for pregnancies, but it's not really written in the sense of being written for a contraceptive. It's written for any drug.
Especially a male contraceptive.
You as the great scientist that you are would make an argument as to whether there's any question at all about safety, either in the partner or neonate or fetus, and then you would have to argue how you would assess that long-term safety.
Yeah.
Mm-hmm. Okay, Diana.
All right, let me pick up then where you left off. And I think there will be some general comments that we can do together. So let me make sure that I'm doing this correctly. I think you have to stop sharing.
I already did. You're sharing now. Just click and push return. It should be the next slide.
Okay. All right. Good. Great. Okay. So this is, as you can see, the traditional valley of death, where you have to get from your basic research into a clinical research setting. And we know what that pipeline looks like in the process for women, because there are a lot of products out there. So there's a history, there's a pathway to follow. But for men, we don't really know what that is. And I think the biggest challenge that I always want to ask Lisa about is, how are we going to develop something when nothing has been developed ahead of us to follow as a guideline? And the FDA hasn't published specific requirements for this method, or this type of method. So many of the questions you're going to ask, we ask all the time, and we don't have answers for it because we just don't know what the answers are going to be. So we know we have to do the basic research, identify the drug, and the toxicology has a very high bar of safety. We need the FDA to address how high that bar of safety is going to be, because we have guidelines for women, but the clinical studies in normal volunteers, how many subjects and for how long? If it were women and it's 20,000 cycles for a new chemical entity, that's about 2,000 women using a product for a full year. That's a pretty large study, and generally they want 2 of those, so 2 of 1,000 each. Is that going to be required for men? And since men don't have cycles, are we just going to use, months, or how? We really need answers to these, and I don't have answers to these, but I'm not sure the FDA does, because they haven't been confronted with accepting or refusing a product yet. So this is the pipeline, the general pipeline. We know all the discovery projects, the grants and the validation, the proof of principle, the medicinal chemistry, and then just getting to the preclinical tests that Lisa mentioned. Some of them are very short to get into the optimization stage, and then the IND. Now this part has to have GLP preclinical tox, which stands for good laboratory practices. And so what you do in the lab to demonstrate safety may not be acceptable to the FDA in some cases. And I was just having a discussion with someone this morning about, well, they did this copper release in a lab, and it's a lab that's sort of set the standard, but will that be acceptable? Because that's not a GLP study. And I don't know the answer to that. And we're always terrified to ask the FDA something, because if they say no, then what do we do? Then you have to do a lot of the toxicology with stuff that's been made under good manufacturing processes. Then you need to scale that up, which is oftentimes a struggle. Once you get the formulation and you prepare the IND, you have to do the first in human clinical trials. Now, in some studies, I mean, I'm not sure how this works outside of contraceptive development because I haven't done product development outside. But what it means for us is we start with a very low dose of a product that's never been tested in humans. And we do about half of the dose group with some placebos mixed in. And we wait until they recover. And if at least part of the group has recovered without any problems, then we do the second half of that group and can move to the first half of the next higher dose. And then stop again and wait for 2 to recover. And then again, finish the second half of that dose and do the first half of the next dose. You can imagine that this is a lot longer process than other studies might be. And that's how we've been told we need to do it. That's how we've done it so far in our single dose first in human clinical trials. After you successfully do that, you'll do a repeat dosing, which is generally a 28-day study. So for that, you need toxicology to support that 28-day study, which means you have gone now from a 14-day or a 7-day tox study to a 14-day or a 28-day or a 3-month study. And then, as you get successful studies there and you want to go into a longer study, you need to do the tox study that will support a longer period of time. So there's a cycle that goes here. You don't have to do a 1-year tox study to support the first in human trial, but you probably have to do at least a 9-month study to get to being able to treat someone for a year. Generally a 6-month study to be able to treat for 6 months. And it has to be in 2 species, 1 rodent and 1 non-rodent. And FDA seems to love rats for some reason. They think they're the most sensitive, so they always want a rat study. So this is the slide that tends to depress people, and this is on some sort of an automatic timer that I didn't realize, and I don't know quite how to do that without the timing on it. So let's see if I can get back. Well, maybe I can just leave it up without going through it. But so what you have is a developmental project, and this is actual timeline of a product that's been in development now for a time. Nesterone and testosterone gel. So it's a transdermal method, and it took about 2 years to get it into the first in human clinical trial. And that was in 2005. And we got to 2012, which is a pretty long time to be able to get into the phase 2A. That trial was very successful, 6-month study of sperm suppression and in about 100 men. And at the end of that, we were ready to go into a phase 2B, which would be an efficacy trial. And it took us about 4, 5, 6 years. And the question is, what were we doing during that time? So we had to do a reformulation to scale up now. We had to do bridge studies. Original study was done with gels that were 2 separate entities, and now we had to put them together and show that by putting them together and making the volume much smaller, we didn't change the properties. And you have to scale up the manufacture and do longer term safety studies. So all of that takes a while. And then we're now in a phase 2B, which was going along swimmingly before the COVID hit. And now we can't follow folks up or recruit new people because the in-person visits are very difficult to have. So we're in a little bit of a suspended animation where we've gotten very creative, and the FDA has gotten an EMA, because this is an international study, have been actually very accommodating with letting us make allowances for how to do these visits. So we're about to get my cat in the process here. In any event, it's going to take about 4 years to do that study, and then if it's successful, then we have to do the phase 3, and that's where we really need the FDA to give us some guidance about how we get from point A to point B, which is the NDA preparation. And at the end, I have a lot of pictures of people that's covering it, but I've estimated it's going to be about 2030 to 2032. So you'll often hear, and every time you do an experiment in the lab that's really successful, the press will say, one step closer to a male contraceptive, maybe 5 years from now, we'll see it. The reality is 5 years from now, you'll be lucky if you see the first in human study if what you've shown is in a petri dish or in mice. Mm-hmm. And now you're going to try to get into humans. So that time frame is ridiculously optimistic, and I think sets up a false sense of security or a false sense of expectation in the community that, oh yeah, we're going to have something in 5 to 10 years that's going to be on the market and that's going to make the difference. I don't think it's going to be that short. Now, maybe I'm wrong. Maybe the FDA has a fast track that I don't know about, but I don't see it happening in a time frame that's shorter than other drug development, which, unless it's a cancer drug, pretty much is about 15 years minimum. So that's all I have to say.
Diana, it's Lisa again. I just want to make a comment, and I want to put a shout out for the MCI initiative that, you know, I hear you saying, oh, we're going to have to ask for FDA's guidance, but I would really like for this group to think about what is the correct way to go here. What would be the appropriate phase 3 study? You know, how many men are partners or whatever the method is. You know, it could be something used in the woman against the sperm, but how many subjects would be included, so that rather than asking and getting FDA's guidance, more telling them what you believe is the correct process and way to go and the correct clinical program, and get their agreement. I know there's always this call for guidance from the agency, but they know less than you on this topic, and they're just going to be as conservative as possible if asked. And you're much better off putting together a really clear discussion of why what you believe is appropriate is the correct phase 3 program, and get their agreement, if that makes sense. They can always have a public meeting on the topic and try to make sure the experts have made their views clear. You know, but any kind of open-ended question will get a bad reaction.
Thank you. So what you need to do then, you're suggesting, is have a product, take it to them in a, there's a term for it when they promise to have you to discuss a product development pathway or approval pathway that they're then locked into for the most part.
Well, sure, you have an IND in the place that you were talking about when you're done with phase 2. You know, if you're an individual sponsor and you don't want to have a big public meeting, you would have a pre-NDA or pre-DLA meeting and describe to them what your plan is for your phase 3 program and why it's adequate to determine safety and efficacy, and ask for their agreement. Because asking them for what they think is just, I think, gonna years and years and years.
But I've watched them. What I've watched them do with, say, Annovera Ring, which was a process that really isn't quite as new as a male contraceptive might be, is that they request a lot of additional studies that you wouldn't have thought you had to do, a QT interval, or, you know, blood pressure, ambulatory blood pressure, and things that are not really something you're anticipating in your development pipeline. And they just request it or say they want it. And I don't know that anybody has there's no experience to be able to fight back and say, well, you didn't require that of product X. Like,
no, you're absolutely right. And as you said at the beginning, there's no precedent here. So yes, you're definitely going to have to do the usual stuff, including the stuff they're asking, like QT prolongation questions and things, unless your animal studies support that you don't have to. But what I'm saying is, you know, rather than saying, you know, you need 20,000 cycles in women's contraceptive development, you would decide, well, what do you think is the right database for efficacy and safety for whatever method you're talking about for a male-directed method, and present that to them. But at a end of phase 2 meeting, if you say this is our program and this will support an NDA, they will remind you of things that they want: drug-drug interactions, QT interval
Right.
prolongation.
Return
They
to
will remind you,
fertility is
even
one of the ones.
if you say, we believe that this program will be adequate to support safety and efficacy, they'll definitely jump in and say, well, yes, but, and they'll give you the list of things they also need you to respond to.
Lisa and Heather both, this is Connie, just to follow up on that, I mean, I think to emphasize what Lisa started out with, I think this is where MCI can really play a role in pulling together a focus group who could say, we got a blank slate here, and here's how we're looking at it. Let's have that conversation with the FDA all the way back to the early, preclinical development sort of phase, and give them those things to react to rather than say and tell us where to go. I think we could do that.
Will they react to it, though, specifically if you don't have a specific product for them to work on? I think they won't.
I don't know.
We've tried to get that general guidance, but it's been not useful.
Yeah, it's pretty impossible for them to comment on a nebulous, especially this division. You could try to do like a patient-focused drug development workshop, because it's an unmet need. But those, I don't think those give you the kind of specific guidance you're looking for. And I agree with you that using MCI's expertise to say, well, here's what makes sense. Here's what's really good science and based on reasonable, approaches, and having a specific, you do need a specific product to get a real answer. Because
right.
Yeah, absolutely. I totally agree with you. They're at least a hypothetical product.
Well, let me ask you this. So if maybe a compromise then is to have it as a workshop amongst people that MCI brings together who could theoretically produce something like this, and just would the FDA allow anyone to sort of be in the audience, not to be put on the hook for anything, but just to keep a line of communication open? I mean, make it a workshop, not make it a presentation to FDA.
Well, there are public meetings. There's open public meetings. Either there's Part 15 meetings, and there's something called patient-focused drug development, where you could ask. I mean, it's a whole process about getting picked for those topics. I mean, maybe the Office of Women's Health could assist in putting together. We did like a gender. I'm looking at my books because I have a publication from one of the gender ones we did in the 90s. I don't know what you mean exactly by, I mean, of course you can be in the audience at a public meeting, but it doesn't really give you a direct communication line with the agency. Unless you're the holder of an IND, you don't really have a direct communication line.
Maybe we can have a separate brainstorm about this particular topic, because it seems like we sort of keep coming back to it. This is new, and what does the FDA want us to do? And then realistically,
well, if you have an actual product and you can ask that question, that's a lot easier than a nebulous.
Right, and maybe we have something in development somewhere we could have that. I don't know, but I think we're on the same page about needing to introduce that. It's just, how do you push that rock up the hill, without waiting a whole lot longer.
So I'll tell you that once upon a time, when I was having the future of male contraceptive meetings, and I asked the FDA to come to the meeting and give us sort of a general guidance on what we're going to need for a male product. And I got a response that was sent to me accidentally because it was supposed to be internal, but it came to me as well. And the comment was, well, we better try to figure this out before we go and talk to anybody else, because we don't have any idea, essentially. So I think it was, at least at that time, now this was back in 2004. So wow. I don't know that they have any more idea now, because I don't think they've had to confront anything in a phase 2 setting for a male method yet.
Right, and they're not going to want to take the time to have a big public workshop if they don't have anything on their plate.
Right.
You know, a sort of, you know, having a discussion about something that they have no need to make a decision about gives them nothing. There's no, you know.
So when you say big public workshop, are you talking about something FDA sponsored or FDA initiated? Because I'm not. I'm talking about more of an internal.
Like if MCI had a meeting?
Correct. If MCI had a meeting, kind of like San Francisco last year, but with one single focus and just one single goal of what to produce, kind of thing.
Well, the question is, what do you want to ask them? Do you want to ask them how to get from the preclinical into the clinical trials? Because that one's pretty straightforward, and they will give you answers on that.
I think Diana, what I'm looking at is not something from FDA, but something to give MCI's grantees and researchers in this arena that gives them a little something more to point at, that's a little more tangible than, oh my God, it's going to take you 12 years. You know what I'm saying? I'm thinking of something that's more helpful to scientists as we, develop drug profiles, targets, and things like that. You know what I'm saying? So it's sort of like, okay, there's all this massive ambiguity, but let's narrow it down to a little something and kind of give them some comfort that it's a direction they can follow. I don't know. Does that make sense?
So now, while I'm listening to this, there are, of course, the testosterone and the related compounds that have been in clinical trial. So why don't we do it baby steps there and perhaps have a workshop just really telling us what was required to do the studies that were done at Washington University. Diana, you were probably involved in some of that, right?
Well, so we were definitely, I haven't been involved in the approval process for TU and some of the products that have been approved for hypogonadal conditions. Again, anytime you have a approval or a product that is targeting as a therapy, then you have a condition you're trying to fix, and that risk-benefit ratio is a little easier to calculate. When you're talking about, in this situation, treating a man to prevent pregnancy in another person, it's a little bit of a different risk-benefit calculation. And I don't think we have a prototype to evaluate it, but certainly it's an argument we've made to get into the phase 2 efficacy trial, where we're now treating couples who are exposed essentially to a risk of pregnancy while they're taking the drug that we're giving them. So we have that experience, and getting to that was a big milestone. Then the question is, and we will at the end of it, if we ever get to the end of it, have the results that we can take as an end of phase 2 to discuss a pathway to approval, which is where you want to be to try to get those specifics. And I think we will, as you said, Gunda, we will use the testosterone approval pathway to prepare what we think is the appropriate pathway to approval.
Yeah, and so I was thinking, while we all work in this space, but we haven't really sort of like heard about it. So that's why I was thinking that maybe that could be a first step in the sequence of conversations to really get people who are, sort of, I guess, working in this space to at least get to know about that particular approval process for, first in human. And so that could be sort of a starting of a blueprint on how we, I guess would go. And then, of course, as you said, the later phases and so forth are maybe not as well developed. But we're going to, we're so far away from anything, probably. And so if we at least know what that very, very first step is going to be, and then, I guess, be a little bit more knowledgeable about that and take that into consideration, for our thinking and planning. And
I
so
think
I'm not
you could easily get some people together who can help you guide you to the first in human study, because there's experience with that.
correct.
Yes.
Well, and that's something that we could potentially put on the docket for a future even webinar in this series, is to start having people walk us through that. And to everyone's point, and thank you everybody for mentioning it, but I just want to reiterate, this is exactly why MCI wants to have this conversation with you all. We want to be able to start preparing or doing what we can to help. So while it may not be, it's been really helpful to see that, because I think that what we've been thinking bigger picture is trying to start reaching out to the FDA without a specific product, more general. So to hear that that's probably not going to be a good path. However, I'm wondering if there is value in us as a community coming together and starting to outline what we think is reasonable. And, then folks can, if we had this sort of core offering that folks, as their products came closer to, the regulatory approval stage, could build on, or, just sort of a general idea or a guidance from the experts of what we think would be reasonable. So that's something we could also consider. But I really like the idea of maybe trying to get someone involved, and Diana, maybe you can help us with that as well, in the testosterone approval pathway, to do a follow-up webinar on this to get us started.
So yeah, I think one of the things we could do that would probably be most useful for you is to have our CCTN investigators at UCLA and UW, who have both put in an IND successfully for a new product that has gotten through first in human and into at least 28-day and sometimes beyond. So we've gotten over that hurdle, and that's the hurdle most of your grantees are looking at this point,
Right.
and guidance on. So I think perhaps a workshop on what goes into that IND and how to get to that first in human trial, what that first in human trial looks like. I think we have the expertise that could and the experience that could, contribute to that. It would be useful for you, I think.
Mm-hmm. I think that sounds great. What do other people think? Does that sound like maybe something folks would be interested in? Okay.
Right.
Debbie, you're on mute.
Still on mute.
There you go.
Sorry, I hate these things. I think it's a great idea. I think most of the people start out in the lab in a basic science context have never thought about any of those kinds of things in any detail. So I think the more kind of education we can have early on would really help kind of at least think about that, even if we're far away.
That's funny. So now we're just done. It's like, well, okay, now, well, we know what we need to do for our next webinar. So,
well, and Heather, Heather, there's a whole range of, I mean, better than anyone, there's a whole range of things that have to sort of happen here. It's not a one-off webinar. It's not a one-off series of Q's and A's. It's going to become sort of an evolving stream of work that I think MCI can really take a leadership role in. Serve a lot of community, service a lot of the community in different ways, one way for the scientists, one way for, FDA interactions or regulatory experts down the road, et cetera.
I would suggest that you do two phases. One is what is the preclinical tox package you need to get the IND started? In other words, that would be working with our toxicology folks at SRI who help us prepare the IND. And that will list all the preclinical studies, the duration, what needs to be GLP, what doesn't, what needs to be GMP, what doesn't, and so forth. So that's that one is a relatively easy recipe to come up with, because the guidance for that does exist.
So Diana, we probably would also need to know what the costs are, because these are, when the things are done under GLP, they become really quite expensive. So that would be maybe something to add as well. totally in detail, but, sort of ballpark figures about all the costs, because that's what the PIs have to consider also. And that's maybe where MCI could be, in the future be helpful, because that's usually not in the grants, and so.
And it's hard to get, it's not considered innovative, so it's never easy
that's right.
to grant for. And it's why the Contraceptive Development Program, or the Contraceptive Development Branch, came into existence, was to provide that service essentially to get from point A to point B for things that you can't really get a grant for. Right. So, and that theoretically should still exist, although I'm not there anymore, so I can't tell you how much it does exist. But the cost is able to be estimated, because again, we've done it, so we know how much it costs to do it under our guidelines. The second part of it is the actual, what does the clinical trial look like? And that's the second, I would say that would be a separate question that you'd want to know once you get past that preclinical package, and because you're going to put a protocol into it and say, okay, now I want to test my product. What does that protocol look like? Again, that's our CCTM people would be really useful, because we've developed those protocols and executed them.
Thank you. That would be great, Diana. Thank you very much. Sorry, Connie, go ahead.
No. I get a little excited, about thinking how we could take this on. But when I was just going to comment that when you talk about the clinical trial side of things, what I think that sometimes forces people to do is think again about their early phase and pre-human kind of work in terms of answering and as many questions as possible then until they get to this clinical trials properly.
I guess real quickly, I want to see, does anybody else have any questions before I do have, I don't know, is everybody familiar with the CCTN that Diana runs? Because that might be a nice overview, because it's really a fabulous program. Would you mind giving just a high-level overview, Diana, of the network?
Okay, so what we have on the male side, we've had this on the female side since '96, and that's when I first got into this field and started working with the clinical development for products for women. The male was introduced into the CCTN in 2004. And so the purpose there was that we had developed back, and by we, I mean people before me in the contraceptive development branch, had developed some progestogenic androgens, molecules that are derivatives of testosterone or nortestosterone, basically, that have been modified to introduce a progestogenic function in them. And that means that one molecule can hit two receptors, essentially. And the progestogenic function knocks out the gonadotropin pathway, stops the testosterone in the testes, and shuts down sperm production. But the androgenic part of the molecule maintains all the androgenic functions that are necessary for ejaculation and other aspects of it. So the premise there was that this molecule delivered either orally or as an intramuscular injection would stop sperm production. To test that, we tested that in animal studies, and we got through the preclinical tox package to introduce it as a first-in-human study. And we developed the protocol, submitted it for the first escalating dose, and that's where I described the pathway where you have to give the first dose and wait for recovery and give the second half of that dose and so forth. It's a much longer than I think your typical first-in-human studies are. But we have, the Clinical Trials Network has two sites that are the sort of what we call the main contractors, who one is at LA Biomed, that's called Lundquist Institute. The other's at the University of Washington. And they've been doing clinical trials in men for years with that same premise of using a progestin, a separate molecule, a progestin, as we're doing in the gel study, where nesterone is the progestin to knock out the testosterone production in the testes and stop sperm production, and then replace with testosterone gel, which is available commercially to keep all the normal functions going, but not so. So I guess the premise for that you need to know is that the testosterone in the testes tends to be 100 to or greater higher than what's in the serum. And you need that high level in the testes to make sperm. So if you lower that testicular level down below a certain threshold, you don't make sperm anymore. But if it's low in the serum, you also can't do other functions that you would want to maintain. So you have to keep the serum levels at a certain level that's functional for everything that androgens need to do in the body, but it's not high enough in the testes to restart spermatogenesis. So that's the concept of how one does this hormonally. So those two sites who've had a lot of experience with this are pre-qualified to bid on doing a new product that we had developed in the branch. And so they began the process of creating the IND to be able to get the first in human trials. And that was successful. And now that first product is able to go into phase 2A trials. In other words, sperm long enough that we can actually show sperm suppression, because sperm suppression in a hormonal method requires treatment for at least 8 weeks or longer to be able to because the sperm that have gotten past the point can still make it through the rest of the maturation process and out into the body. Now, if you have a method that can work faster than that, you may be able to get away with showing effectiveness with shorter term studies than we can do by a hormonal method. So, but the CCTN has the capability of looking at a product and saying, okay, we can do this first in human trial. We'll figure out what endpoints we need to look at. We'll write the protocol. We'll submit it to the FDA. We'll put together the package. At the same time, we have 2 support contracts that can do the GMP manufacturing of a product that's not available commercially. And we have a biological testing facility at SRI that can do the preclinical tox, and they have the capability to do that. So if we have our scientific advisory committee approve a candidate, then we can test it through some of these toxicology pathways and pay for that, and then get it into the clinical trial. So the CCTN is the clinical component of that, but the contraceptive development program is sort of that whole package of recognizing a promising compound, trying to help support the the preclinical tox to get it to a clinical trial, and then designing and carrying out the phase 1 trial. Does that make sense? Does anybody have any questions about that?
I have a general question so at what point would investigators approach you to start that process?
Well, our thought, we're all in a kind of a new world of this, because the program used to all reside in what's now the contraception research branch. And we separated out the clinical part of it and those 2 contracts and put it into the intramural program. So the idea would potentially be, if you have a compound that is considered very promising and it gets funded through some of the grant programs that CRH is still funding, potentially they can bring it to our attention. And we can potentially submit it to the scientific advisory committee and see if they're enthusiastic about it enough to bring it into our pathway. Now we can do that independently of the CRH, but the process for it is not incredibly well defined. And so, I don't know exactly how we would do that. Generally, I could just ask the investigator to present it to the scientific advisory committee to rule on and, make a determination and all. But how much money we would be able to then invest in it, it might have to get into a queue, because we do have a lot of things that are already kind of in the pipeline. So that's not a really clear answer to your question. But we don't have like an RFA. We're not extramural, we're intramural. So we don't have an RFA that we can put out that you can apply to.
Yeah, I think, again, any kind of information that helps people understand what exists and what approaches you need to start a process is useful for the community. That's all.
All right. Any other questions? This has been a great discussion. Going once, we're 2 minutes over.
No, just a comment. Thanks to both of the leaders here, Lisa and Diana. And Lisa, I find your very to the point PowerPoint slides very comforting personally. So
yes, thank you very much.
People from my era understand.
Yeah, I get it. I love it. It's all good.
Yes, very much. Thank you, Lisa and Diana, for taking the time.